Hi, everyone, and thank you for joining us today for a webinar on turning FFPE challenges into sequencing wins, where we will be discussing a dual nucleic acid extraction solution from the same sample. My name is Claire McLean, and I'm a marketing associate here at Omega Bio-tek. We will be joined today by Sarah Amiramati, one of our field applications scientists, as well as Lauren Golan, an associate scientist with our R&D team. On the agenda for today, I will start off with a brief introduction to Omega Bio-tek before handing you off to Sarah for a review of the challenges of FFPE extractions, as well as our automated solution for DNA and RNA co-purification. From there, Lauren will review some performance metrics and downstream sequencing data that we have collected. We will end the session today with a Q&A, so be sure to submit your questions below. With that, I would like to introduce you to Omega Bio-tek. We were founded in 1998 and are based out of Atlanta, Georgia. Our expertise is in DNA and RNA purification solutions, and we boast a portfolio of over 400 products encompassing different extraction technologies, including magnetic beads, silica spin columns, and filter plates. Omega Bio-tek is ISO 9001 and 13485 certified, and our presence is felt around the globe through more than 50 distribution partners. At Omega Bio-tek, customer success is our top priority. We understand the challenges of achieving high-quality DNA and RNA purification in a timely manner and work to remove these obstacles by making quality, automated products and workflows more accessible and affordable. By focusing on collaboration, innovation, and integrity, we empower our customers by providing manual or automated optimized solutions designed with them in mind. With that, I will hand it off to Sarah for more about our FFPE solutions.
Thanks, Claire. Hi, everyone. My name is Sarah. I'm a field application scientist here at Omega Bio-tek, and I'm going to walk you through the updates we've made to our Mag-Bind FFPE DNA RNA kit. Formalin-fixed paraffin-embedded, or FFPE tissue, are widely used in clinical and research settings for long-term tissue preservation. As a result, it represents the most common source of archived clinical material available for retrospective studies. However, these samples present unique challenges due to the fixation and embedding process, which can cause cross-linking and degradation of nucleic acids and proteins. This affects the quality and reliability of downstream analyses. Deparaffinization is also an additional hurdle, and traditional methods often rely on toxic solvents like xylene, adding safety and disposable considerations for labs. So how do we address these challenges? We've updated our Mag-Bind FFPE DNA RNA kit. It's a purpose-built solution for co-extracting both DNA and RNA from a single FFPE section. Starting from as little as one 10-micron FFPE scroll, this workflow delivers DNA and RNA into separate eluates. No sample splitting is required. This means you preserve precious material while gaining access to both genomic and transcriptomic data from the same section. The chemistry is designed with challenging samples in mind. It uses xylene-free deparaffinization and a formalin scavenging lysis buffer that efficiently reverses cross-linking. The result is PCR-friendly nucleic acids recovered in roughly 5 hours of total processing time. Because this kit is magnetic bead-based, it's fully compatible with major automation platforms, including Kingfisher and Hamilton Microlab Star, so you can scale to high throughput processing without sacrificing consistency. While we've updated the protocol, all the buffers used remain exactly the same, so there's no change to the chemistry itself. It's the same reagents you're already familiar with. What we've done is optimize lysis of the samples. Previously, this involved a 4-hour incubation at 56 degrees Celsius. In the updated protocol, we've reduced that incubation to just 1 hour by implementing a 90 degrees Celsius incubation step followed by lysis at room temperature. This allows us to achieve comparable results in a fraction of the time. So let's walk through the dual extraction workflow. There's a short xylene-free deparaffinization, which can be done with HistoChoice, HistoClear, hexadecane, or mineral oil. The tissue is then rehydrated and softened in a formalin scavenging lysis buffer. An optimized 30-minute room temperature proteinase K digestion occurs, followed by reverse crosslinking at 90 degrees Celsius for 1 hour. Again, because this kit utilizes magnetic beads, the bind, wash, and elute steps can be performed on most open liquid handlers and magnetic processors. DNA only or RNA only extractions are also available for flexibility. We have a method designed for the standard Hamilton Mag-X star configuration, using only Hamilton components with no third-party integrations required. The deck provides ample space for tips, plates, reagents, and includes 4 Hamilton heater shakers for incubation steps, 4 magnetic positions, and waste management for both single channel and MPH pipetting. After obtaining the lysate, the method processes up to 2 plates in parallel, 2 for DNA, followed by 2 for RNA, using both the 96 head and 8 independent channels, with a total throughput of up to 192 samples per run in 2.5 hours. Now that we've walked through the workflow and automation setup, I'm going to hand it over to my colleague Lauren, who's going to take you through the data.
Thank you, Sarah. Hello, my name is Lauren Golan, and I'm an associate scientist here at the Research and Development Lab. And today I will be walking us through the functionality data where we will be showcasing our Mag-Bind FFPE kit's performance along with 2 competing kits. Now let's get into the data. What we wanted to understand here is pretty straightforward. How does our Mag-Bind FFPE kit perform across FFPE sample types? And how does it compare to what's already out there? So we used 3 tumor types, breast, lung, and colorectal. And for each of those, we paired the FFPE with a matching fresh frozen control as a reference. And then we compared our kit against 2 commonly used kits. Our column-based competitor here, a company Q, and then a magnetic bead-based competitor here, company T. And for this assessment, we used NanoDrop, Agilent Tape Station, qPCR, and sequencing. So starting with the yields, our DNA is comparable or better in breast and lung when comparing to company T, our bead-based competitor in orange here. And we are comparable in breast to company Q, our column-based competitor in blue here. Looking at our RNA, this is where we really shine. So we're seeing statistically significant high yields across all 3 tissue types. So up next, we're going to look at eluate purity using the NanoDrop. So at a high level, the 260-280 absorbance ratios look pretty similar across all kits, all close to that 1.8 range, which is what you would expect for DNA. And then looking at the 260-230 ratios, company T in orange stands out as performing below Omega and company Q, indicating contamination in their DNA eluate. So then moving to RNA here, looking at that 260-280 ratio, all of the kits are around that 1.9 range, with an ideal RNA ratio being more of 2. This is not unusual for FFPE as a highly degraded sample type. But looking at the 260-230 for both competitors, we can see the ratios are lower for our competitors, indicating contamination. So with us in green here, we're performing higher. This suggests Omega has cleaner eluates overall, which becomes more relevant when you start to think about PCR efficiency and sequencing performance. So here we compare our functional performance using PCR. All of these samples across the board were diluted tenfold. And as we can see, the DNA here is performing very similarly across tissues and kits. Where we see a big difference again is in the RNA, where we're seeing lower CT values with our extraction here in green. This indicates more amplifiable nucleic acid, which lines up with the significantly higher yields we saw via tape station in the last slide. So now looking at RNA quality using the DV2 analysis. For heavily degraded FFPE RNA, where a tape station RIN or RNA integrity number would not be suitable, we look at the DV200, which is simply the percentage of RNA fragments over 200 base pairs. It's basically telling us what percentage of fragments are still long enough to be useful in sequencing. And what we're seeing here is Omega's DV200s are consistently above the 70% mark across all tissue types, indicating high quality RNA. So here we are comparing DNA and RNA CT values back to a fresh frozen baseline for each tissue type. All the nucleic acid was normalized by mass. So all of these values here that you are seeing is independent of yield and speaks to the quality of the nucleic acid extracted. So the delta CT values being closer to 0 indicates that the FFPE samples more faithfully reflect the original gene expression profile. So again, the goal is 0 here. For DNA, we're seeing everything being comparable across the board. But with RNA, here again, we stand out. We are getting closer to that theoretical 0. So up to this point, we reviewed yield, purity, and PCR performance. Let's walk through how this translates into sequencing results. Here we're going to zoom in on breast and lung tumor FFPE. So let's dive into the whole exome sequencing data. For Omega, metrics like insert size here and target coverage here are trending closer to the fresh frozen control for breast, as can be seen by this 173 value compared to this 219. And then also in lung, we're seeing that Omega is close to the control here. Also looking at the target coverage, things are looking pretty comparable across the board, and with the aligned reads also similar, so no major concerns here. Next, we conducted a concordance analysis, which is basically asking how similar are the variants we call an FFPE compared to the fresh frozen. So we decided to display this with the Jacquard coefficient, which is a representation of the similarity in variant calls between the baseline group, our fresh frozen control, and the test group, which is our FFPE. In other words, how many specific variant calls were made in both the control and test sample runs relative to the total number of variant calls made. So just to walk through this calculation, first we'll look at the intersection or variant calls found in both the control and test samples and divide that here by our union, which is the variance only found in either the FFPE or the frozen control. So when these two are divided, this gives us a measure of similarity where 1 would be identical and 0 would mean no overlap. So just quickly glancing at this data, we can see here that in breast, Omega seems to have the highest similarity, the coefficient that is closest to 1, whereas for lung, things are more comparable. And we'll see this visualized in the next slide, where we have a Venn diagram really displaying this intersection and union. So here we can see that in breast with our high 0.77 coefficient, this indicates we are recovering more biologically relevant calls while introducing fewer artifacts. And then also taking a closer look at lung, the story is more comparable across the board, with all kits performing at a coefficient of about 0.74 or 5. So transitioning to the RNA-seq side. So we look fairly comparable looking at the insert size here and at the average percentage properly paired reads. Where we see the big difference is here when we look at the duplicate reads. So we're seeing company Q stick out with this 63.7 and with this 43.5. With that being nearly double in terms of the duplication rate. And that matters because the duplicate reads don't add new information. They're essentially taking up sequencing space without increasing usable data. So even if the total reads look similar, higher duplication can reduce your effective depth. And then looking comparatively at Omega's metrics, they typically point to better input complexity. So you're getting more unique fragments rather than repeatedly sequencing the same template. And then here we compare our RNA quality looking through the lens of differentially expressed gene analysis. A DEG analysis shows the count of genes with the expression levels significantly different than the fresh frozen control baseline here, shown either upregulated in green or down in gray. So in both tissue types, but especially here in lung, Omega has the lowest level of both up and down regulated genes relative to this control baseline. And this indicates the expression profile produced by the FFPE samples processed using the Omega method more closely resembles those of the more biologically relevant fresh frozen control. And this kind of harkens back, and it's likely due to Omega's vastly superior RNA quality, thinking about those absorbance ratios, the DV200s, and also the superior yields. So in summary, Omega Bio-tek's Mag-Bind FFPE DNA RNA kit can extract high-quality DNA and RNA from the same FFPE sample. We see strong performance in RNA yield and quality for sure, but without compromising the DNA. And because this is a magnetic bead-based workflow, this is well suited for automation and higher throughput environments that is advantageous over a column-based strategy. So thank you for your attention. We have some time for questions, but if you'd like more information on anything you've seen today, you can visit our website where you can request a free sample of the Mag-Bind FFPE DNA RNA kit, or see our app notes for further details on the data presented today. Back to you, Claire.